Welcome to Detours, Understanding Acquired Brain Injury. This is Unit 2, Chapter 3, Putting It Together. So let's talk about how codons are used to put together to build proteins. So when a gene is to be read to make proteins, they have to be decoded first. From DNA as it is encrypted in a sense, because genetic information in DNA is in a compressed form. Those of you who've opened compressed files know it has to be decompressed first, and then it can be worked with. So we only have 30,000 or so genes, and this is plenty though due to the way it's compressed. DNA, as we decode it, you need to produce RNA. Remember I talked about coding RNA, whose job it is to convey the information. So we need to make there are different types of RNA that we need to make in order to transmit this information. You also need a structure, those of you who remember, called a ribosome. Actually, you'll need a bunch of these because usually you're going to make more than one copy of these in order to put together a protein. So many copies will be made, but we'll just look at one in order to understand the process and understand this happens a whole lot at once. So there are several types of RNA, and we need a few different types. First, we need to make an RNA known as messenger RNA. Messenger RNA, a coding type of RNA, will convert the DNA into that readable form. All right, now remember how I said that DNA strands run anti-parallel to each other. So they need to be decoded in the opposite direction in order to produce that protein in the correct direction, so that it's not a misfolded, because misfolding leads to serious errors. Okay. When the cell is producing a specific protein, these events will occur inside the nucleus first. All right, so first we get what's called transcription of messenger RNA transcription. Step one is initiation. A special type of RNA called RNA polymerase, try saying that 5 times fast, works almost like an engine. It'll move along and it will bind to a segment of DNA called the promoter. If you remember, I mentioned the promoter first. It says like start here. It's that initial flag telling it where to start on the DNA. Clunk. Then the polymerase, the RNA polymerase, will start there. And it literally then will begin to travel along that segment and begin to copy. So the RNA polymerase finds that marker and it will move the length of the DNA as it is unwound with the help of the histone, and it'll start attaching, click, 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 complementary nucleotide sequences. These are brought in to the nucleus and they're also found around. As it unwinds from free-floating RNA segments that are produced earlier, throughout the cell's lifespan, because they have to be ready to perform this function. So it will begin to pull apart with the help of the histone, and the RNA polymerase will begin to assemble the messenger RNA in a process called elongation. Here's a diagram. And it will put together a single-stranded RNA of whichever side of the DNA it is attached to. When the RNA polymerase reaches the terminator sequence, there's a specific marker on the end which tells, on the end of the DNA, of that gene it is copying. Remember, these genes are there to make specific proteins. So it tells the polymerase, stop here, disconnect, and at the end of the elongation from marker to terminator sequence as it puts together the entire messenger RNA sequence. Remember, it's in reverse because nucleotide, nucleotide. So as it reads, it will put the opposing either, thymine to adenine or cytosine to guanine. So click, 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 all the opposing letters to it. And when it hits the terminator, it knows to detach that messenger RNA. That marker tells the polymerase where to stop. Finally, there's the termination and post-processing for this messenger RNA to build this protein. All right, non-protein segments are going to be clipped and removed. So anything in this gene that you're using to build this protein, there are other messenger RNAs whose job it is to cut and remove any parts of the gene which aren't needed to actually make the protein physically, which are control segments, for example, telling it stop, start, anything like that are cut out by matching it and snip. Finally, then it'll be capped for its ends, polymerate, so it is ready to leave the cell. So a little bit of final preparation. Then it'll leave the nucleus through a nuclear pore on hole or opening in the nucleus. They're little tiny, and it will find its way out of the nucleus because it's now prepped. And it will work its way into a ribosome. But still not ready because, great, you've got an RNA, a messenger RNA already to make a protein. We just now built a control RNA. But now we have to, because we had to decompress. We're done decompressing the information from the DNA. Now we're ready to take that decompressed information and make it into a protein. All right, so what happens next? The messenger RNA finds its way to a ribosome outside in a rough ER, rough endoplasmic reticulum, or there are free-floating ribosomes. So it depends on where it's going. It depends on what the protein does. So what the protein is going to do, that it's going to make, where this will go. Again, a lot of this has to do with like electrical charges on the messenger RNA and what kind was unlocked. It will go based on its chemistry and its charge. It'll be attracted to certain places. Much of this is still not well understood. We study this in microscopes, under microscopes and computer models and in silico, as that's called. In vitro in the laboratory, in vivo in cells. And so, okay, so the new messenger RNA finds its way to a ribosome, and it's a template in effect. It is a mirror template, reverse, to build this protein. Now, the ribosome is what actually builds. It's the factory for protein. If you remember Unit 1, ribosome is made of two separate pieces. The mRNA binds to what's called the small subunit, S30. And the large subunit, the other piece of the ribosome, or S50, will bring the amino acids together that are in the cell to the start segment of the mRNA, and it will literally travel the length of this messenger RNA in a process somewhat similar to what happened to build, but there are definitely differences. All right, the initial binding of the mRNA to a ribosome is called initiation, just like happened before. The S50 component of the ribosome is responsible for the catalysis, different ways to pronounce it. Anyway, and the small subunit, S30, is responsible for the decoding of the mRNA. All right, so elongation. As the combined pieces of the ribosome, the whole ribosome moves along the mRNA, reading these codons. Remember, it reads in groups of 3. this structure from outside is called transfer RNA. Now, transfer has different codon groups, okay? It may be like ATT on the end, or GCC, or CCC, ATT, or TTT, or, whatever. It has those 3 different nuclear bases on its end. And as the ribosome moves along, it will bring in, based on their charge. Again, this drives a lot of this, is the charge and the chemistry of these different transfer RNA. Their name tells you what they do. These structures are cloverleaf shaped. They have on their ends amino acid groups attached to them. And so the tRNA has the opposing set of codons that would match with the mRNA. Now, not all tRNA, of course, match with what's on the messenger RNA. It's inside, that's now kind of clipped to the ribosome. And so only those that would fit as the ribosome moves along will be plugged in to the messenger RNA. Meanwhile, on the opposing end are the amino acid groups, and they start to build up, linked. And the enzymes will assist sometimes in making sure the amino acids link up because they're connected to the tRNA. And eventually you get this long polypeptide chain building up because it's not finished yet. And it gets longer and longer and longer and longer and longer as more and more, as the ribosome attaches more and more transfer RNAs to the messenger RNA. Click, 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 putting it together. Eventually, and this is called elongation, just like before. Termination. Eventually, the ribosome will chain enough of the amino acids together until it reaches the stop codon at the end of the messenger RNA. Then it will cut, and the amino acid chain will be released. That gets severed, and clunk, the messenger RNA is set loose. It's done its job, and now we have on the other side long amino acid chain. So what matters most is the chain of amino acids. The tRNA and the messenger RNA are kind of used up now. It is the tRNA can be rebound and all that, but they're kind of, what matters is that polypeptide chain of amino acids altogether. And they get help from the enzymes and other, they either may be linked to another amino acid chain, or they may fold on their own, or they may get help by themselves. The newly synthesized chain of amino acids, which is polypeptide chain till it's formed, folds based on the charges of the different atoms that make them up. And they form a very complicated three-dimensional structure that is capable of performing many different components in the cell. It's not done yet, though. Other modifications may be needed. Enzymes may need smaller proteins or metallic or metalloid coenzymes to help them. They may need to be cut. They may need more folding afterwards. They may need to be packaged by the Golgi apparatus, or may need to travel through the endoplasmic reticulum, or they may need to be put inside a vesicle, something like that. Finally, after these final modifications, then they're ready as proteins. Now you've got your protein. So what's it going to be? It could be almost anything that the cell needs. It could be part of the cytoskeleton, or So this is how, maybe it's a new tRNA. Maybe it's going to be part of a ribosome or whatever. Obviously, this is a very simplified description of how genes make proteins. We'll discuss a bit more in detail, but yeah, this is the process. This is how. It's quite a bit to it, and it is repeated again and again and again and again. The cells are amazing, and this happens millions and millions and millions and millions and millions of times per day inside your body and mine. Obviously, this is a simplified explanation, and we will stop here. The next episode, we will talk about cell division, the process known as mitosis. This is not the production of sex cells. This is about asexual cell division. Please click like and subscribe. Don't forget to hit that bell icon all if you want to receive our information. If you have any questions or comments, please post them below. Hopefully see you next time. Please share this with anyone you think may benefit. Leave any questions you may have below, and see you next time. Bye.